this became feasible, and then with the advent of next generation sequencing, this can really be done on a massive scale. So if you have unmethylated cytosines and you treat DNA with sodium bisulfite, then you will convert all unmethylated cytosines to uracil. However, if the cytosine was methylated, then it will still remain methylated after the bisulfite conversion. So we've got uracil, which will then subsequently behave as a thymine in PCR reactions. However, methylated cytosines will still remain a cytosine. So if you want to do bisulfite in informatics, it's actually rather simple. Let's assume we have a sequence like this, and we have two residues here and here that are methylated. We convert the DNA with sodium bisulfite. We do a C to T conversion of all the unmethylated residues, as you can see here. However, the methylated residues, they remain cytosines.
Felix, we lost your voice.
Right. I'm very sorry. So where did I stop?
After 10.
Okay. Okay, and I probably said so. When you bisulfite convert the sequence, then you convert all unmethylated cytosines with thymines, but the methylated residues remain Cs. And then once we map this to the genome, and we know exactly where this sequence, which is called a methylation call, and then we can simply determine was at this position, was there a T or a C in the read, and therefore we can say this was unmethylated, 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 and this one here remains methylated. So this is the theory of bisulfite sequencing. However, it's not quite so simple, but I'm going to show you this in a little bit. So just generally, the bisulfite-seq analysis workflow is you get some sequences of an Illumina sequencer. So this is really dependent on very good quality sequences. So at the moment, it's really only Illumina sequencing. Then there will be some kind of a processing pipeline, and this is what we're going to talk about this morning in good detail. And then in the end, you will do some kind of methylation analysis. And what we're going to talk about today, excuse me, is all the steps involved in the initial processing pipeline. I want you to get to understand. What is that? It is pausing screen share. Can you still see me, the screen? Hello?
Yes, I can see you.
Okay, sorry, there was some kind of message. I want you to understand what your data is and what it looks like, and understand all the processing steps before you actually start doing the methylation analysis. Right, so when I said it's actually quite easy, you just do a C to T lookup from the bisulfite converted sequence and the genomic sequence. It's not quite so simple, and I'm trying to illustrate this here. So if we assume this is the genomic locus, this is the top strand, this is the bottom strand, then we do the bisulfite conversion, and that's done on single-stranded DNA. So here, all the unmethylated residues are converted to U, and methylated residues remain C, and we do this separately for the top strand and for the bottom strand. And now afterwards, we have to perform a PCR amplification to get double-stranded DNA back. And this would then look like this. For the top strand, we now get a sequence which we call OT for the original top strand. And as you can see here, in comparison to the original genomic locus, the sequence looks different because of the conversion. Here in red is the original top strand, and here in red is the original bottom strand. And now by PCR, we also regenerated complementary strands. So CTOB would stand for complementary to OB and complementary to the top strand, marked here in blue. And now you can probably appreciate that all these 4 sequences we are now getting is different in the sequence to the actual original genomic locus. And this is a problem because because we need to align, or we need to find out where the sequence came from. And the problems here are that for any given single genomic locus, we now have 2 different PCR products, and we can, in theory, also have all 4 different sequences, so the original top strand, the original bottom strand, and the complementary versions as well, in the mix of what comes off a sequencer, and we need to find out where it comes from. And then, to make things even worse, each of these 4 sequences can theoretically exist in any possible methylation state. So we can have this residue methylated and this, or this, and so on. So it's really quite a difficult task. And so what we are doing is 2 things. First of all, we are converting all sequences in silico and perform an in silico bisulfite conversion, which means that if we have a sequence, we just simply convert every C to a T, so that it doesn't matter which one was methylated and which one wasn't. We just assume they were all unmethylated. And we just do this for the mapping step, and in the next slide we can show you that how we get back to the real sequence. But this is to get around the problem that we can have every single conversion state. And there's another thing which we're doing. So this kind of approach is called a 3-letter alignment of bisulfite seq reads, because we just get rid of every cytosine and convert it to a T, and then we're left with the 3 other bases of the DNA alphabet. But as I showed you, we can have different sequences for the top strand and for the bottom strand, and therefore if the sequence would come from the top strand, we would need to do a C to T conversion, which is this one. The converted residues and T's are marked in red, and for the bottom strand as well. And the bottom strand would also be a C to T conversion, but that's equivalent to doing a G to A conversion on the top strand. So this is now starting to get a bit confusing. Okay, but we need to do this for the top and bottom strand separately. And we also need to perform this C to T conversion of the top strand of the genome, but we have to do this only once. And we also need to do a C to T conversion of the bottom strand, which is equivalent to a G to A conversion of the top strand. So we need to have 2 copies of the genome, which are bisulfite converted in silico, and then we index those so we can align against them. And we also need to convert the sequences either for the top strand or for the bottom strand, and then we need to perform up to 4 different alignments. So C to T converted to C to T converted genome, to the bottom strand, and then this sequence to that. So we can end up with 4 different alignment threads per sequence in order to be able to identify where a sequence came from. I know this is probably quite complicated, and it catches me out as well sometimes, but it does work. And then the next step in this procedure would be to find out where is actually the best alignment from any of those up to 4 genome alignments. And once we figured out what is the best alignment, we then know its position, and then it's easy. Then we do the easy part. Then we extract the genomic sequence from that position, and then we do the methylation call and look up whether this was methylated and wasn't. And what we then are actually interested in is this so-called methylation call string, which is being output by Bismarck. So that's the software we wrote to do this back in, I think, 2010. And In this methylation call string, we encode various different bits. So whenever there's a dot, it means it just involves any base that is not a cytosine in the genome, so it's something that's irrelevant. Then the sequences can be in different contexts. So Z is the CPG context, which is most relevant for mammalian systems. And then there are also two non-CG contexts. H would be CHH. In this case, H stands for every DNA base, but not G. So for example, this one CAT would be a base in CHH context. And X would be a base in CHG context, like this one. If this was a C, CG, that's CHG context. And then in addition to that, we also encode the methylation state in this string. If the characters are lowercase, the base was unmethylated, and if it was uppercase, then the base was methylated. So this is actually what all this is about, and what the researcher wants to get out of a bisulfite dataset. Right, so we use Bismarck to do this, and I just mentioned that you can have up to 4 different strands. However, it's in most cases it's actually not so bad, because normally we deal with so-called directional libraries, and this depends a little bit on which kind of adapters you used and which protocol during the library preparation. But normally you only get the original top strand and the original bottom strand, and you never ever see the other 2 strands. That's really good, because in that case you only need to perform 2 alignments and not 4. There are, however, also different protocols. So in this case, the PBAT, that's a technique that has been developed around 5 years ago, and I'm going to talk about this in a little bit later, because it's really good for low cell and input material. And in these cases, you only ever see the complementary strands, CTOT and CTOB, but not the original strands. So again, also in this case, only 2 alignments, which is sort of okay, and not 4. And then there are also so-called non-directional libraries, which is where you can really have all 4 of these strands, OT, CTOT, OB, and CTOB. And this is really the most computational intensive mode you can think of. You need to have 4 alignment strands in parallel in order to figure out where a sequence came from. And there are a couple of different commercial kits that make use of this. The most notable is probably the Zymo picomethyl kit, and also the single cell bisulfite seq protocols. They are also based on some kind of pre-amplification of the material. And that means that we also get these 4 strands. If you can, then probably avoid this technique, because it's quite computationally intense and also doesn't allow you to look at certain things, but overall it still works. Right, then just to say that the alignment works in this case. If you look at the blue line, which we call biased here, this is a normal 4-letter aligner, in this case bow tie, and you can see that the mapping efficiency is a function of the read length. So the longer a sequence is, the more alignment you get. But even with 25 base pairs and a normal DNA sequence, you get around 80% mapping efficiency with simulated data. However, if you do a 3-letter alignment, here called unbiased, then you can see that really very short reads, they have a very poor mapping efficiency, and this is just because we reduced the letters in the DNA down from 4 to 3. However, if you increase the read length to 50 or 75, so back in the days when I started, we normally had 36 base pair long reads. They were around here. You can see that they are actually not very different to using 4-letter aligners, and right now we're probably more in the 100 to 150 base pair region. So it actually, the mapping efficiencies are not much worse than for normal alignments. So that works. And here you can see simulated data, excuse me, simulated data where I simulated different methylation levels in all three contexts, CpG, CHG, and CHH. And you can see whatever we simulated, it was really exactly found like that with Bismarck alignments. And also the mapping efficiency is pretty much unaffected by the methylation state. So whether it's highly methylated or poorly or intermediate, you will always get the same methylation state, the mapping rate. So this works. Now, again, we're going to talk a bit more in a bit more detail about the QC trimming and mapping steps. And then after the question and answer session, we talk a bit more about methylation extraction, some different means of doing quality control after mapping, and then you can proceed doing your analysis. So I'm not quite sure how much you know about sequencing. I'll just keep it very brief. This is a screenshot of a FASTQ file, which is there are 4 different lines per sequence. The first line is a sequence ID field. The second line is the actual sequence. The third line is irrelevant, and the fourth line is a quality control string, which corresponds exactly 1-to-1 to each of the base calls. And this is a measure of how accurate the base call was from on the Illumina machine. So these files are quite nice, but they can contain up to 1 billion lines, and they are in a text file, and they are very unwieldy, and you can look at a few sequences, but it doesn't actually tell you very much. With the advent now of the NovaSeq, these files can probably go up to 40 billion lines. So we need some kind of means of dealing with this data, and Simon Andrews in our group, I believe he's also going to give one of the next webinars in the coming weeks. He wrote a quality control tool called FastQC, and this really, within a couple of minutes, is going to tell you an awful lot about your experiment, and especially about your bisulfite-seq experiment, as I'll point out in the next couple of slides. So you can see a couple of things for your record keeping, file name, total number of sequence length, and these kind of things. But there are also very important measures and statistics, like the base call qualities, potential contaminants, the base composition especially, and which kind of duplication you have to expect from your data. Not sure if you're familiar with FastQC, I'll explain it anyway. On the x-axis, you can see the position in a read and base pair, so from the start to the end. In this case was a 100 base pair long read. And on the y-axis, you see the quality of the base calls in a Phred score. And the Phred score translates into an expected error rate per base call. So 30 would be an error rate of 0.1%, 20 is 1%, 10 is 10% error, and so on. So roughly speaking, you can just look at this. At the traffic light system, if the sequence is in the green part, it's good. Yellow is and then as soon as things start going below 20, you probably want to remove the sequences and be left with a good portion of the sequences. Then a very, very cool plot in FastQC, which is not so helpful for other kinds of sequencing, is the base composition plot. And here you can see all the 4 bases adding up to 100%. And you can see that cytosine in this case for a whole genome shotgun bisulfite sequencing is very low, in the range of 1 to 2%. And at the same time, the levels of T is elevated. And this is a result of the C to T conversion of unconverted cytosines. So with a single glance at this graph, you can already see that your experiment has worked really quite well. If you run different kinds of experiments, for example, RRBS is a quite common type. This stands for reduced representation bisulfite sequencing, and is a result of digesting the genome with an enzyme called MSP1 to start with. this enzyme cuts at a CCGG sequence, and that means that all sequences you want to see in your experiment should start with either CGG if the base, the first C was methylated, or TGG if the base was not methylated. And this is exactly what you can see here. So 60% is here CGG, and T would be TGG in 40% of cases. And if you add these numbers up, then you know already that in almost 100% of cases, the sequences actually start at an MSP1 site. This is a very good quality control tool, which you can say very quickly that your experiment has worked really beautifully. There's also a duplication rate plot in here, which not only tells you how often you saw some sequences, so these are the ones that were unique 2 times and so on, and here are sequences that were present 10 or more times, 50 or 100 or more times, and so on. And it also tells you a rough number if you were to deduplicate your sequences just based on the sequence right now and not on the mapping position, then you would probably have 20 or 30% remaining. So this sequence we're currently looking at is around 70% duplicated. In some cases, you would expect a very low duplication rate, for example, whole genome shotgun sequencing. However, there are certain other applications like RRBS or amplicon sequencing, target enrichment, these kind of things, that these are actually applications where you would expect a higher duplication rate. Again, this plot can very quickly show you if you see that your experiment is 95% duplicated and you didn't expect that, then you already know that something went wrong. It also tells you whether there were overrepresented sequences in your library. This plot shows every sequence that was present in more than 0.1% of the total library. And here in this case, you can see various different ones that all differ by maybe 1 or so base pairs. And you can see here that most of them actually come from Illumina PET and PCR primer. So this is a contamination. And if you add up these numbers, then you will already very quickly see that this adds up to at least 35 or so percent of or 40% of all your library. And this, you can also use this to modify your expectations. So if you then see a lower mapping efficiency later on, then this is not necessarily a problem, because you know that 40% of your library is actually Illumina primer contamination. So therefore you have to adjust your expectations. Then there are also common problems in bisulfite-seq, and this is actually probably the same for all kinds of other sequencing, but this becomes really quite apparent only in bisulfite-seq, because you do get a decline of the overall quality towards the end of a read, and the longer the read, the poorer the qualities normally. This is one problem, and another problem is also that you can see that the amount of cytosine starts to increase the longer your sequences are. And we have figured out that this increase, there is no biological reason why there should be more methylation. More methylation towards the end of the read. And this is actually adapter contamination. So whenever you read through your read and read into the adapter on the other side. And this is something you cannot spot in normal sequencing or in not bisulfite sequencing, because their C and G levels are exactly the same at around 20 to 22%. And there's a flat line. And if there's adapter contamination, you can't see this. But because here our levels of cytosine are so low, we can actually see this increase. And this is something you need to remedy before performing the alignments. This is a screenshot of some data we pulled down from the short read archive, and it depicts a library that has actually quite shocking quality scores. If you look at the per sequence quality score here, and if you look at the quality scores per sequence, you can see that there are some sequences with a fairly good overall quality, but there's also an awful lot of sequences here that have a very poor quality score, of in this case 2, which is an additional, it's a flag by Illumina saying, please do not use these sequences, we don't trust the sequence ourselves. And there is a way around this, so even if you get data from the short read archive, you can quality trim the data, and then this is before and this is after quality trimming. Admittedly, we did lose 50 or 60% of all those poor quality sequences here, but there is some data that is actually worth salvaging, and the remaining data in here is actually of quite good quality. This is also just a warning. Not everything that's submitted to the public read archives is really good quality data. It often, more often than not, is actually quite surprising what people submit. This is the same kind of data, and again showing from the start of the reads to the end, there's a huge increase of the methylation level, if you will, of cytosine. So the level of cytosine creep up to almost 25% towards the end. But if you look at the adapter contamination plot in FastQC, you can see that from around 30 or so bases, we start reading into the adapter as a read-through adapter contamination, and this is actually causing this increase in cytosine because the Illumina adapter is methylated. So therefore you start, or it actually has a C content of 50%. This is why you get this this increase. Again, if you trim sequences with adapter trimming in this case, then you can detect and remove the adapter sequences, and then you can see that in the salvaged portion, the levels of cytosine are now really quite low, and this is data that we can align to the genome. Also here in this plot, the adapter contamination has left. People often ask, why is there this blip or this strong increase of T's at the very last position and the dip in A's? This is a result of very stringent adapter trimming. We don't really know whether a sequence is actually adapter at the end of a sequence or whether it's a genomic position, but if we can't be sure, then we rather remove it. And the Illumina adapter happens to start with AGA-TC. So if the last base of any sequence is an A, it could be a genomic A, or it could be the first base of the adapter. So we trim it off. And that is the reason why there can actually not be an A at the very last position of a read, because it might be an adapter. Similar to AG, sequences won't end in AG and then AGA and so on. So this is something we have to accept in order to not have any adapter contamination, because reads are just not going to align very well, and we're using very stringent mapping criteria, as I'll point out later. So I think this is the last slide, and then if you want to, we can have a quick question and answer session after this. We think it's very important to trim sequences before you align them to the genome, because if there is any kind of adapter in the sequence or any kind of sequence that doesn't belong there, then you will have a low mapping efficiency, and this essentially translates into a waste of money. So you want high mapping efficiency. But even worse, if the sequences get misaligned, then the misalignments, they result in errors, and the errors will be almost 50-50. You either call a base a cytosine or a methylated cytosine. So it's just really a gamble. So you really want to avoid misalignments, and also adapters need to be removed because they are methylated, and as soon as they are in the read, you will start making erroneous and wrong methylation calls. Right. Do you have any questions at this point?
Okay, thank you very much, Felix.
Well, if you if you don't have any questions, then I can also continue. I'm happy either way.
I got some questions for you.
Mm-hmm.
Can you use the Bismarck for the 3 different library like directional or PBAT and non-directional library?
Yes, so we've got the default mode in Bismarck will be the directional mode, where we'll just use the OT and the OB strands. It also has an option -PBAT, where it will only do the other 2 alignments, the complementary ones, and the option - non-directional will do all 4 alignments. But you can later serve to the GitHub page, or I can link the website of the manual of Bismarck. Maybe I can put it in the chat here, and there you can see all the different options. There's by now quite a lot of documentation of how everything is done, and we describe a lot. Let me find the link.
Okay, thank you very much.
Thank you, Felix.
Yeah. Any other questions?
I'm wondering. We saw your PowerPoint file, and actually, in our class, we downloaded your program and run the sample test.
Yeah.
And it gives some M-bias plot.
Yeah, that will be part of the next session.
Oh, okay. Next. Okay.
Okay.
Sorry.
You should go ahead for your presentation.
Actually, we used your program to run to analyze the publicly available methylation data, RRBS data. So next time we will ask some more real questions based on our output. Some of the part we can understand, and so we can maybe ask you more technical questions later.
Yeah, but I think maybe we just continue with the next part, and then we can ask your questions.
Okay, great.
Okay.
Okay.
So I pasted in the link to the Bismarck user guide, which explains most of the things. Hold on, maybe I can share. Where is it? The entire screen. So this is the user guide. On the right-hand side, you can see the table of contents, and you can also click here and go to the sections directly. Here is a section which is really quite useful if you want to look at which kind of sequencing do you have, which kind of trimming do we recommend, which kind of mapping do we want deduplication or not. And also there's then some kind of additional explanation of all of the different things, what is affected, what affects the M-bias for the different kits, cymomethyl, Swift, random priming, and all these things. And then at the end, there should also be a fairly comprehensive list of all the options, which you can look up here, or just go on the command line and type whatever program you're using and --help, and it will show you the same thing. So this is really quite useful. Quite useful link.
Okay, thank you. So the next presentation will be November the 3rd. Song-hee?
No. Just Felix will continue to on next presentation about the mapping the so DNA methylation calling and these things.
If you have a few more minutes, then I would just continue now for the second part.
교수님 계속해서 좀 한 30분 정도, 20-30분 조금 더 하고 이제 퀘스천 받도록 할게요. 예.
Okay, thank you.
Yeah.
Okay, well, in that case, so that it's not too late, I'm going to just move on now.
Yeah.
Okay.
All right. Good. So we were done with the adapter trimming now. The next part will be once you've got really good quality data that, okay, hold on, get rid of that. Once we have good quality data, we need to align the data, and that can be done with Bismarck. And then the output of Bismarck will normally these days be a BAM file. A BAM file is it contains much more information than a FASTQ file, as you can see here. So this would be a paired and read, where the first box in dark blue is read 1, and the lower box would be read 2. They're actually on one line each, but it doesn't fit on the screen.
Felix, we cannot see
Felix, we cannot see your desktop.
Yeah.
Again, sorry?
We cannot see your presentation file.
See your desktop.
Presentation.
Okay.
Yeah.
There we go.
Good. Now it's fine. Yeah.
Good, there we go.
Yeah.
Okay, so here's the read 1 of a paired end file, and here's read 2. So a BAM file contains more information. So it is still embedded here is the sequence in this line and the quality score, which we also had in the FASTQ file. But now we also know the mapping position, on which chromosome did the read align to, and where did the read start at which position, and a few more things. The most important one is probably the methylation call. This one, the XMZ flag, where you can see these dots and whether or not a base was methylated. And this is then actually the data we are interested in. Yeah. So these files contain much more information. However, they are still not very easy to use, because they still contain hundreds of millions of lines. And in addition to that, it's a binary format. Just a quick word on sequence duplication, because this is quite relevant for most parts of bisulfite sequencing. If you have a very diverse and complex library, then you can see here there are many different reads should start at many different positions. And if we have a read that's actually a PCR duplicate of another read, then they would start and end at the very same position. But this is a quite complex and diverse library, and here you would not lose a lot by deduplication. If you have a very duplicated library, on the other hand, then you would only have very few positions that are actually unique, and many reads that start and end at the very same position. And this often happens when you have very low starting material, so you start amplifying everything up with PCR. And we would normally recommend deduplicating these libraries if it was a genome-wide sequencing assay, which would in this case collapse all the reads that are exactly identical down to a randomly chosen one. So you can see that you will lose quite a lot of reads in this case, and this can also mean that the methylation levels change at certain positions. But still, this is something you should probably be doing. And then people normally say, yeah, but if we deduplicate, we lose a lot of data. And we argue that you actually don't lose data, because the data you had before was not actually real data. It was simply the same data amplified, artificially blown up. So even if you have well, if you deduplicate the data, you can still cover each position or each cytosine by as many reads as your reads are long. So if you have 100 base pair long read length, then you can cover a given position up to 100 times without them being deduplicated. And this is recommended because it's very, very unlikely that you get exactly the same fragment of the same length starting at the same position twice from several different cells, and it's much more likely that it's actually a PCR duplication artifact. However, there are also different applications where deduplication is absolutely not advised. For example, RBS is one of these. As I mentioned before, if the reads start with CCGG on both sides, then you really only expect all sequences to start at this very position. And if you then were to deduplicate this data, you would end up with one read for this position, and that means you would discard more than 90% of your data, and you really don't want that. Similar thing is with amplicon data or target enrichment. Okay, so this was just mentioning duplication of the data. So then we would take the BAM files that are produced by Bismarck and extract the actual methylation call data. Again, here I'm showing read 1 and read 2 of the example of the BAM file 3 slides ago. And you can see read 1 starts in this direction and read 2 would go in this direction. And in the middle, there's apparently here an overlap. We also, we don't want the reads to overlap because the methylation calls are produced by PCR and they are therefore redundant and don't give you any additional information. So what we do is we ignore this overlap and we extract the methylation data from BAM files using Bismarck methylation extractor, and by default it will disregard or ignore the overlap of reads. In this case, we're always taking all the methylation calls from read 1 because they tend to be of higher quality than read 2. And if we take read 2, as soon as read 2 starts overlapping with read 1, we stop extracting the calls. Then by default, we are normally only interested in cytosines in CpG context, so marked here in these shaded boxes. We would then just extract the actual positions of all these individual cytosines and end up in the primary output of the methylation extractor, and this is the CpG methylation output. This is now reduced to a simple text file with 5 columns. This is the read ID. Then we have a methylation state column where plus means methylated and minus means unmethylated. Then we have the chromosome and the position. And in this case, it's only a single base per position because we're looking at cytosines at single base resolution. And then at the end, there's also a context saying whether it was CHH or CpG context. So these files are much more useful than a BAM file. However, the problem is, as you can see here, there can be many different positions per read. So these files can be even longer, maybe up to 20 billion or so lines, so still very unwieldy things. But we can take this CpG methylation output and convert this into a more useful format. And we've got a script to do that, and this is called Bismarck to bed graph. So what this step is doing, it will read in all the different positions and cytosines in this file, and it will then sort them first by chromosome and then also by position within the on each chromosome, and then end up with either a bed graph or, even more useful, the coverage output, which now encodes the chromosome, the start and end position, and this is at the same position because this the format is just, again, single base resolution, but is a format requirement. Then the column 4 will be the percentage of methylation, so 100%, 60%, and so on. And then in columns 5 and 6, we would have the number of counts that are methylated and unmethylated. And this is now actually a very useful format because the number of lines in here equates to the number of cytosines that was actually covered and seen in your experiment. And there are up to 45 or so million CpGs in the human genome. So if you have a number of 30, then you know that you covered almost 60% or so of all possible cytosines or CpG cytosines in the human genome. This brings down the number quite noticeably, and this is also a very good format to do further analysis in. For example, using R or any kind of other software. This is really useful, and you can also submit it to Geo or so as supplementary file, which they require for a data submission. So this is already a good output, the coverage output. If you want to, you can even take the coverage output and further transform it into a genome-wide cytosine report, a CpG report by default. This will take the coverage output as input files and then output a file like this, which with chromosome, the position, and then in this case, the strand is really the true strand of that position. So this would be a C on the top strand, top, bottom. and in this case there's always a pair, top and bottom, and then it will give you the number of methylated and unmethylated calls, as well as the dinucleotide and trinucleotide context of these files. So if you have several different experiments and you want to have a matrix with all possible calls from all samples that is really the same in all conditions, then you can generate this program. So this lower report, the cytosine report, is based on the genome. It will actually only have, or it will have one line per C in the genome, whereas the coverage output is every cytosine that was covered in your experiment. So these are two different things, and this means that the genome-wide report can have many more lines than the coverage report, and it will even report lines where you have no calls at all, so 0, 0, unmethylated or methylated. So this file is a little bit inflated and contains, I think, around 1.5 billion lines, but it really is very comprehensive. And it also has many different options these days. For example, you can merge the top and bottom strand Cs into a single dinucleotide entity if you want to do that. We have additional filtering options for GNOME-seq data if you want to assay methylation as well as accessibility at the same time. So do look at the options there, but there are quite a number of options. But anyway, either of these two outputs should be suitable for pretty much anything you want to do from there on. Right, now we're moving on to the second last part, which is mapped quality control of your data. And this is something you can actually really only do after the data has been aligned and not before, like what FastQC is doing. So this here is, as you mentioned before, the so-called M-bias plot, which is a methylation bias plot. Again, we're looking at these from the start to the end of a read, and follow for each position in this example, we follow the average methylation at this position. And what we would like to see is, it shouldn't matter whether you have a methylation call at the start or at the end of the read or in the middle. You would always expect the same average methylation calls. And here's a little bit of a wobble at the start. This is probably a technical issue, but overall this is looking really fairly nice. It's also a good opportunity to look at the non-conversion, the bisulfite conversion efficiency. So if you look at the CHH and CHG context, you can see here that they are methylated very, very lowly, maybe around 0.5%, which is really good. And it also tells you that the bisulfite conversion efficiency must have been at least 99.5 or 9.7% if it doesn't go any higher than that. If you see the non-bisulfite, the non-CG methylation around here, about 20%, then you know that something went wrong with your experiment, and maybe you want to repeat the experiment. It also, this plot can also show you things like that. This is now read 2 of a paired end sequencing, and you can see that overall the methylation level is fairly constant at around 65%. However, right at the start, there is a steep drop, and at the very first positions, the methylation looks like it's very, very low. And again, there is no reason why there's no biological reason for this. And we figured out that this is a problem of the end repair and A-tailing process. So if you fragment the DNA, then you have to fill in some of the ends with cytosines in the lab, and for this you normally use unmethylated cytosines because it's simply cheaper. If you fill this in and then perform the bisulfite treatment on these residues, then they will appear unmethylated. And if you sequence this read like this, you will also sequence these at some point if your read is long enough. However, if you do paired end sequencing, then you sequence the bottom strand of this, and in this case, every single read you sequence will start with an unmethylated C, and this is exactly what we see here. So this is an artifact which you cannot spot unless you look at the M-bias plot, and is certainly something you need to do. For For alignments, you should really ignore the first couple of positions of read 2 of paired-end sequencing, because they will introduce artificial data. And this is what we do by default at our institute, and you should be doing this as well. It might be a little bit different if you mainly do RBS data, but I'll get to that in a second. Okay, so ignore the first couple of positions. There's an option in the methylation extractor, -- ignore read 2, and then give it a number, and then you should be fine. So here's this is a similar case for RBS data, which is illustrated here. Again, this is the MSP1 site, CCGG. The enzyme cuts like this, and then all of your sequences would start with CGG or TGG. This is exactly what we saw already in the composition plot. Also for RBS data, you expect a really high duplication rate, because there are only a couple of 100,000, maybe 600,000 sites in the genome where you would size select these fragments. And that means if you sequence 50 million reads and you only have 400,000, 500,000 fragments, then you will start sequencing each and every fragment several times. So therefore, you get really really quite high duplication rates. But for RBS, this is fine. Also, just to mention that RBS reads, this is an in silico digest of the human genome, showing that the MSP1 fragments are actually really, really short. And this is because of the CGGG recognition motif, which is mainly present in CPG islands. And if you want to, if you do this digest, then you can get a lot of really, really short reads. In addition to that, you normally perform size selection between 40 and 220 base pairs, and then there are not that many fragments that are actually longer than that. So if I sometimes get emails from people who did RBS and they sequence with a 2x300 base pair protocol RBS reads, and this is completely pointless, because as you can see here, the majority of reads will be very, very short. And if you sequence longer than that, then you sequence the fragment, and then you sequence into the adapter sequence. And so the vast majority of all reads will only be adapter. And if you then on top of that do paired-end sequencing, you sequence the second strand, which has been already covered completely by read 1. Again, in the other direction, so this will all be ignored, and you will waste a lot of money, and it will take much longer to analyze. So think about which kind of fragment size distribution you have. Normally, the fragments in bisulfite sequencing are rather low and peak around 100 to 150 bases. So really super long reads overlapping paired end don't really help. Often it's enough to have one fairly long single end read. Also in bisulfite, in RBS libraries, we do get these artificial methylation calls, similar to what we saw in paired-end sequencing. So if you digest the genome with MSP1 and then fill in this position marked here in red, this position is filled in every single RBS fragment, and it's unmethylated. So whenever you sequence this cytosine, it will have a wrong methylation call. It will always look unmethylated. And similarly, if you do paired-end sequencing, then this sequence will look unmethylated as the second base of every experiment. So that needs to be treated. And we've got a wrapper around CutAdapt, which we're using. It's called Trim Galore, and this has specific options for RBS data with the option --RBS. It will take care of this. Whenever it finds adapter sequence on the 3' end, it will trim an additional 2 bases of read 1, and of read 2s, it will remove the first 2, so that you never ever have these contaminating artificial calls in your data. And this is, as I mentioned before, the so-called PBAT data, which stands for post-bisulfite adapter tagging. It's a technique that got quite a lot of traction over the last couple of years, because the authors claimed that it's suitable for very low input material. On the left-hand side, you can see the normal bisulfite-seq protocol of whole genome data. where you fragment the data first, you then ligate on adapters, and then you perform the bisulfite treatment. And bisulfite treatment is a very harsh chemical treatment, so it often leads to breaks and nicks of DNA, and those fragments are not going to be sequenced, but you sequence only whatever is left over after the treatment. And PBAT is very different in that it doesn't have any fragmentation adapter ligation at the start, but it uses bisulfite treatment as the cutting and denaturing agent itself, and then you ligate on adapters to every fragment in the library afterwards and sequence everything. And if you want to know how the details are, this is done by using first strand random priming, the original paper with 4 nucleotides. These days people use 6 or 9 base pair reads, pull this down with biotin, and then perform a second strand random priming and second strand synthesis as well. And this is how you then get the fragments with both adapters on it. However, this data has some complications. It has a very pronounced sequence composition bias at the start, and this also translates through right to the methylation, the M-bias plot. As you can see here, this was a 4 base pair PBAT, 1, 2, 3, 4, and you can see that these bases have a very different average methylation, and also there's a much increased average methylation in non-CG context. And whenever you see something like that, it points you to additional QC steps. So in this case, what you should be doing is you should be removing these sorts of biases before you actually perform the mapping. So Trimger has these trimming options for the 3' end, also for the 5' end. The recommendations are in the link I shared with you. Please remove these reads before you align them, because we found out that the reads actually have quite a lot of SNPs, and they also have quite a lot of indels, and as you can see here, they introduce quite a lot of bias and erroneous calls in your data if you leave them in. At this point, it's maybe a good point to quickly mention qcfail.com. I'm not sure if you're aware of this. If you Google qcfail sequencing or go to this website, this is a site which we have started to populate with common problems in sequencing. So not limited to bisulfite sequencing, but all sorts of things. So mapping problems, adapter contamination, any issues really to do with sequencing. And you should probably go and have a look. Some of the more bisulfite specific things that I mentioned here today are also mentioned there. For example, this is the mispriming we just talked about, and there's also this the bias at paired read 2 of paired end reads. So it's a really good and valuable tool shed where you can go and find out about common problems. Right, I think with that, we are probably now at the end of this session today. Just to briefly recapitulate, before we perform any alignments, we suggest you do some kind of quality control with FastQC. Then Trimger Law is a wrapper around CutAdapt that handles all of your quality and adapter trimming issues. There's additional functionality for RBS reads. And if you go to the Trimger Law website, either on the Babraham bioinformatics side or on GitHub, there's also an RBS user guide, which has some additional information about RBS. So once we've got really good pre-treated data, we align the data with Bismarck and get output BAM files. Then depending on your application, the data will be deduplicated, and then we continue to extract the methylation information with the methylation extractor, and optionally further transform this into more useful files, such as the genome-wide cytosine report or the coverage files. And this is now also the step where you can look at the M-bias plots. And only the M-bias plots informs you if something went wrong, and you might have to then actually go back and remedy this after you spotted the errors, but it's still a useful thing to do. And finally, if you run Bismarck to report or Bismarck to summary, this will this will generate graphical HTML reports, and then yeah, you can just click on the link, and they are a little bit interactive, so you can mouse over it with different positions, and we'll get some additional information out. At this Epigenesis website, there's also a protocol where we've written down all the steps, including the commands for the data, but you can also just download the practical for this session, which has pretty much the same data. And then there are some useful links at the end of this presentation. Some of the programs you might or might not use for this kind of analysis. QC fail, there's the link again. And with that, that's the end, and thank you very much for your attention.
Felix, one of my students ran your Bismarck with the publicly available data, which is DNA methylation on mouse to predict age, and he in his M-bias plot, he found that something, the plot looks kind of weird. So he wants to ask you how to, interpret that output. So here, post his output in the chatting box. Can you see the URL? Could you click that and take a look
at the moment, I can only see the code for the HTML file, but not the file itself. Oh yeah, there it is. Never.
Oh, you can download that HTML file and then look at it in the browser.
Okay, so
after you download it.
I've got the file. So is this, can I ask, is this data from the Stubbs et al. Publication?
Which publication?
So Thomas Stubbs should be the first author. Is it the one from our lab?
That's right. That's right.
Let me check.
We use the epigenetic clock data published in the Genome Biology.
Yeah, that should be the one.
Yeah.
So,
okay.
Yeah. So, I think something went wrong there. You started off with 10,000 or almost 11 million reads, and you only have a 1.8% mapping efficiency, and the levels in here are completely weird. So something went wrong, I think, with the processing here. And the M-bias plots look super funky, probably because there are so few reads in there. Just to mention, the protocol that was used in this case is, I believe, read 2 of these files has an 8 base pair UMI at the start, so that's a unique molecular identifier that needs to be removed before you do anything. Then also the reads will have CAGTA as the next 5 bases after the first 8 bases UMI, and that's introduced by the adapter. That also needs to be removed. Okay. And only once you have done that, then you can start aligning the data, and then you need to do a normal adapter trimming as well. So it's a bit more tricky than normal data, and we introduced these UMIs so that we can discriminate between RBS fragments that are PCR amplified and proper unique different fragments. But in this case, I would suggest that you probably just drop me an email, and we can then go through the steps that maybe you describe the steps, what you've done, and then I'll try to respond to that, because it's a bit difficult to just do this in a.
Okay. Yeah. Great.
Okay. We will send you the command we processed.
Yes, that would be good.
Take you.
But I'm pretty sure that we can fix this, and I can send you the script to extract things as well, so that's all fine.
Yeah. Okay.
He also he was also asking whether should we use mouse reference genome version 37 or 38? I think he used
What?
38.
That's a matter
Is
of
that
taste.
some?
Yeah, the mouse genome was.
Yeah, I don't see.
Sorry, but the mouse genome was updated. When was it? In 2012 or 13? So there's no real reason why you should now use version 37. I would,
Okay.
if you start something new, then you should use the latest genome.
Yes, 38. Okay.
Okay, thank you.
Okay, thank you very much for Felix. I think this is time to end of the webinar today.
Okay.
Yeah. And if
Good.
we have the further question, we can email you or send you.
Yep. Anytime.
Yeah. Okay. Thank you very much.
All right.
Thank you very much.
Good.
Wonderful.
Thank you.
Okay.
And have a nice evening in Korea. Bye-bye.
Bye. 네, 교수님.
예, 잘 됐고요. 지금 아마 녹화가 됐을 것 같은데.
네, 저도 이렇게 녹화를 했는데 파일이 이렇게 두 개로 나눠질 것 같아요. 예, 그래서 이렇게 뭐 교수님 거나 제 걸 보고
맞습니다.
같이 합치든가 이렇게 해도 괜찮을 것 같거든요.